Welcome back. We are on single molecule digital sizing of proteins in solution. Courtesy George Craner, Raphael, Matthias et al., Timothy, Gian et al., Quentin, Peter Ivo, Greta Sneijder, Czechalska, Now we deal with single molecule digital sizing of proteins in solution. And to go with the abstract, the physical characterization of proteins in terms of their sizes, interactions, and assembly states is key to understanding their biological function and dysfunction. However, this has remained a difficult task because proteins are often highly polydispersed and present as multi-component mixtures. Here we address this challenge by introducing single molecule microfluidic diffusional sizing, SMMDS. Now we deal with sizing of heterogeneous oligomer populations by SMMDS. A heterogeneous mixture of alpha-synuclein oligomers 10 picomole was probed in a single-step scan SMMDS. Measurement of single molecular burst events were extracted using the burst search algorithm. And to differentiate between differently sized assembly states of alpha-synuclein oligomers, the value for the minimum number of fluorescent photons threshold N-min was varied while keeping the interphoton time threshold constant. This allowed for the equation of burst intensity distributions which differ in molecular brightness. Exemplary burst intensity distributions for 4 different N-min threshold values are shown. Light blue is 5 photons, orange 20 photons, green 30 photons, and red 47 photons. It's visible here in the diagram, right? The insert displays burst intensity histograms in semi-logic scale. Intensities are normalized intensities with respect to burst duration. Exemplary B's exemplary diffusion profiles generated from burst intensity distributions with 4 different minimum number of fluorescent photons. Threshold values panels are color-coded according to the specific threshold values used in a panel. A diffusion profiles as shown in blue lines, experimental fits as orange lines, and error as green bands. The extracted hydrodynamic radii RH with errors are given as insets. For a definition of errors, please refer to the legend of figure 1b, C. The next is C, extracted RH oligomer subspecies displayed as a function of the different minimum number of photons threshold values used in the burst search algorithm. Data represent extracted RH errors as reported in panel B. The colored vertical bars indicate threshold values used in panel A and B. The horizontal dashed line indicates the ensemble average size the entire oligomer population generated from a diffusion profile from all bursts obtained from the measurement. RH of monomeric alpha-synuclein as measured by SMMDS is indicated as a dotted horizontal line. Source data are provided as a source data file. Sizing of multiple species with a heterogeneous aggregation mixture by SMMDS. Many protein systems are heterogeneous. Aggregation mixture by SMMDS. Many protein systems are heterogeneous, multi-component mixtures consisting of proteins and protein assemblies that differ in size by several orders of magnitude. Many protein systems are heterogeneous, multi-component mixtures consisting of proteins and protein assemblies that differ in size by several orders of magnitude. For example, aggregation mixtures are made up of monomeric protein and large fibrillous species. Often one of the components, example the monomeric protein, is present in large excess, while the other one, example fibrillous species, is only present in small amounts. Approaches that can quantify the sizes of such differently populated species are much sought after, yet lacking SMMDS can fill this gap as it has the capability to size molecules and assemblies in heterogeneous mixtures even when an excess of one of the molecular species is present at bulk levels. To demonstrate the potential of sizing protein mixtures, that are compositionally heterogeneous, we set out experiments with a sample system composed of fibrils formed by the protein alpha-synuclein, a key component in the pathology of Parkinson's disease, and an excess of monomeric alpha-synuclein at a nanomolar concentration. Such a mixture is often encountered in assays that probe the mechanisms underlying protein aggregation and amyloid formation. We first performed continuous scanning experiments on a pure alpha-synuclein fibrils at 10 nm monomer equivalent concentration and pure alpha-synuclein monomer at 10 nm concentration to establish the signature of the two species. Notably, within the fibril sample, only 10% of the monomers are fluorescently labeled. Similarly, only a fraction of 10% of labeled protein is present in the monomer sample, thus ensuring concentration parity with the fibrils and facilitating direct comparisons between the two. Sizing of multiple species with a heterogeneous aggregation mixture by SMMDS. And this diagram is a schematic of an aggregation reaction composed of monomeric alpha-synuclein fibrillar species. Red sizing of alpha-nuclein fibrils in the presence of an excess of monomeric alpha-synuclein. And C step scan measurement of a mixture of alpha-synuclein fibrils, 9 nm monomer equivalent monomeric alpha-synuclein. Comparison of extract sizes of triplicate step scan measurements. Pure fibrils of 10 nm monomer equivalent, and top panel show exemplary fluorescence time traces. The simultaneous yet independent measurements of the sizes of nanoclusters and monomeric protein further allows estimating the number of monomer units per nanocluster. This is done during comparison of the volume ratios of monomeric TDP43 and the clustered form. Assuming no restructuring of the protein within the nanocluster, a single cluster could contain as much as 20,000 proteins if the cluster is composed of pure protein. However, as condensates are liquid in nature and contain solvent molecules, typical volume fractions of proteins within condensate systems are on the order of -10 to 35%. Hence, we expect the number of proteins per cluster to be in the range of 2,000 to 7,000. Discussion. The physical characteristics of sizes, interactions, and assembly states of light proteins is vital for a heightened understanding of the biological function, malfunction, therapeutic intervention of proteins. Particular interest are insights of the compositional heterogeneity of proteins and protein mixtures such as protein oligomers, protein aggregation, protein condensate systems. So you have protein oligomers, protein aggregation, and protein condensate systems. However, such characterizations is challenging to achieve using conventional biophysical approaches, as these methods are mostly reliant on ensemble readouts, which limits sensitivity and the information content that can be retrieved from such measurements. The SMMDS approach developed here and addresses these challenges and takes advantage of the high sensitivity afforded by single molecule detection and the physical features of diffusion, the microfluidic regime, to enable digital sizing of protein assemblies, and the heterogeneous multi-component protein systems directly in solution and in a calibration-free manner. With different examples ranging from single proteins to protein assemblies, we have illustrated how the digital nature SMM-based approach enables diffusing based monitoring of protein hydrodynamic radii down to the femtomolar concentration range, thereby pushing the limits of diffusion sizing experiments by more than 5 orders of magnitude. And it's further characterized the polydips nature of proteins is characterized as being assemblies on the example of a protein-protein aggregation reaction and applied SMMDs to discover nanoclusters of a phase-separating condensate system. These examples highlight the capability of the approach to elucidate properties of low abundant species present in heterogeneous biomolecular systems. The next topic will be on interfacial property determination from dynamic pendant drop characterizations. Courtesy to Ziqiang Pan, J.P. Martin-Trostner, Zijun Jin, and Keqiang Zhang, Nature Protocols 2024. Thank you for the abstract, and this article will be of help to so many who are learning about biological methods. So before we can proceed, just want to show you the topics we'll be covering on the latest research and reviews in biophysical methods are articles from Nature Portfolio. The other subjects on coverage will be, we've just finished disposing single molecule digital sizing of proteins in solution. And after the present topic would be the third one, lipid polymer nanoparticles to probe the native-like environment of intramembrane rhomboid protease, GRPG and its activity, cells for viscoelasticity is deformation dependent, and research on a self-saturation nonlinear SSVEP signal feature extraction algorithm, multi-domain interaction mediated strength building in human alpha-actin dimers enabled by direct single molecule quantification. So onto interfacial property determination from dynamic pendant drop characterizations. The abstract. The properties of the interface between materials have practical implications in various fields, encompassing capillary action, foam and emulsion stability, addition properties of materials, and mass and heat transfer processes. Studying the dynamics of interfaces is also fundamental for understanding intermolecular interactions, change of molecular conformations, and molecular aggregations. Pendant drop tensiometry and its extension, the oscillating drop method, are simple, versatile methods used to measure surface tension, interfacial tension, and interfacial rheological properties. These methods can, however, generate unreliable results because of inadequate material preparation, an incorrect calibration method, inappropriate selection of data for analysis, neglect of optical influences, or operating the system outside the linear viscoelastic regime. In addition, many studies fail to report accurate uncertainties. This protocol addresses all these critical points and provides detailed descriptions of some operation tips relating to purifying methods for different kinds of material, the time frame for analyzing measurement data, the correction method for optical effects, implementation of the oscillating method with a common programmable pump, and remedies for some common problems encountered during the measurement. Examples of interfacial tension, measurements for 2 and 3 phase systems, as well as interfacial dilation modulus measurements for N2, and surfactant solutions are provided to illustrate procedural details and results. A single measurement takes minutes to hours to complete, while the entire protocol, including the leak test, cleaning, repeated measurements, and data analysis, may take several days. Well, the key points of this: Surface tension, interfacial tension, and interfacial rheological properties can be calculated from droplet images. These properties are essential for research and application in energy development, material science, biological engineering, and mass and heat transfer. While pendant drop tensiometry and the oscillating drop methods are simple and versatile, their accuracy and reliability require careful materials preparation, calibration methods, data analysis, consideration of optical influences, and uncertainty calculation. Similar content being viewed would be on-chip protein separation with single molecular resolution, direct digital sensing of protein biomarkers in solution, and single molecule mechanical fingerprinting with DNA nanoswitch calibers. So we proceed from SMMDA to the following topics: on-chip protein separation with single molecular resolution, courtesy Adams Rehin, Shiloh, O'Hayon, Diana Hunter, and Emmett Miller. Abstract: accurate identification of both abundant and rare protein hinges on the development of single protein sensing methods. Given the immense variation in protein expressions levels in a cell, separation of proteins by weights would improve protein classification strategies, upstream separation facilities, sample binning into small groups, while also preventing sensor overflow, as may be caused by highly abundant proteins in cell lysates or clinical samples. Here we scale a bulk analysis method for protein separation: sodium dodecyl sulfate, polyacrylamide gel electrophoresis STS page, to the single molecule level using single photon sensitive wide field imaging. Single molecule sensing of the electrokinetically moving proteins is achieved by in situ polymerization of the page in a low profile fluidic channel having a depth of only 0.6 micrometer. The polyacrylamide gel restricts the Brownian kinetics of the proteins, while the low profile channel ensures that they remain in focus during imaging, allowing video rate monitoring of single protein migration. Calibration of the device involves separating a set of ATO647N covalently labeled recombinant proteins in the size range of 14 to 70 kDa, yielding an exponential dependence of the protein molecule weights on the measured mobilities as expected. Subsequently, we demonstrate the ability of a fluidic device to separate and image thousands of proteins directly extracted from a human cancer cell line. Using single particle image analysis methods, the detailed profiles were created of the separation kinetics of lysine and cysteine labeled proteins. Downstream coupling of the device to single protein identifications may provide superior proteins classification and improve the ability to analyze complex biological and medical protein samples. Similar contents are streamlined single-cell proteomics by an integrated microfluidic chip and data independence, 3D projection electrophoresis for single-cell immunoblotting, single-molecule digital sizing of proteins and solutions, which has already been covered. Introduction: single-cell proteomics, sensing, identification, and quantifying the vast number of proteins in a single cell remains one of the most sought-after holy grails in biomedical research. In analogy to the profound impact that single-cell transcriptomics has made in contemporary scientific research, it is broadly anticipated that single-cell proteomics will carry a comparable or larger influence on all aspects of life sciences and medical research. Unlike nucleic acids, proteins cannot be replicated in an enzymatic reaction, that is, polymerase chain reaction. Hence, this limitation, combined with the knowledge that many proteins in a cell's proteome appear as a single or just a few copies, motivates the development of single molecule sensing techniques. Working towards this goal, single molecule approaches based on labeled antibodies, Edmund degradation, fluorescence resonance energy transfer, FRET, and nanopores have recently been developed but have yet to be applied for profiling and anti-proteome. Recent simulation studies of single protein electro-optical nanopore sensing showed feasibility for whole proteome identification based on the labeling of only 3 amino acids, lysine, cysteine, and methionine. However, the vast dynamic range of protein expression in cells further complicates sensing and analysis, and obtaining such high accuracies in practice requires significant improvements in both the spatial and temporal resolutions of single molecule techniques, the protein labeling efficiency, and the development of viable approaches to address the large numbers of protein molecules in each cell. One strategy that may considerably improve the ability to classify and correctly identify proteins involves the controlled separation of proteins by their molecular weight prior to their molecule by molecule identification. Not only may the strategy remove highly abundant proteins such as albumin, representing over 50% of the plasma proteome, in favor of the more clinically relevant proteins, example lighter weight transcription factors, cytokines, etc. It also potentially improves the accuracy of the identification algorithms due to the small repertoires of proteins that need to be matched in each molecular weight band. For bulk samples, conventional SDH page has been proven to be a highly efficient method for size separation of proteins. Furthermore, SDS page has been extended to deal with smaller, albeit bulk sample volumes using electrophoresis-based microfluidic devices, enabling the quantification of selected denatured proteins. A lab-on-a-chip approach its advantages over conventional SDS page due to shorter analysis time, reduced sample consumption, and importantly, better compatibility with downstream single molecular sensing technologies. The controlled delivery and separation of proteins at the single molecule level present a new challenge in terms of both the required photon-sensitive wide field optics and high-resolution microfluidics in which a polyacrylamide gel needs to be embedded, unlike in bulk SDS page in which the fluorescent signal is derived from ensembles containing tens of thousands of proteins. The device should enable real-time monitoring of single protein motion through the fluidic channels. Furthermore, to maintain throughput, hundreds of proteins should be tracked simultaneously, necessitating high-resolution wide-field imaging over single-point detectors. Finally, the device should be compatible with downstream sensing approaches such as more single-molecule fluorescence and silicon-based nanopores, which have been shown to be effective at protein detection and classification. To meet the above criteria, we developed a complete in silicon has been developed, which is it was seen that was a complete in silicon. Let me repeat that. To meet the above criteria, a complete in silicon hard microfluidic device containing a UV activated polyacrylamide gel for single molecular separation of fluorescently labeled proteins was developed. The polyacrylamide gel slows down the Brownian kinetics of the proteins, while the low-profile fluidic design, 0.6 micrometers deep, restricts the proteins near the glass surface, permitting high-resolution wide-field imaging using high numerical aperture in NA optics, and real-time imaging of individual proteins during the entire separation process. Calibration of the device is performed by analyzing a set of proteins in the size range of 10 to 70 kDa, pre-labeled molecular weight, which are shown to separate in less than 3 minutes. The data empirically agrees with an exponential dependence of the proteins' molecular weights on their measured mobility in the gel. Following separation, proteins can be ejected from the acrylamide gel into the separating channel for subsequent processing. Subsequently, we demonstrate the ability of a fluidic device to separate the image thousands of proteins directly extracted from a human cancer cell line using single particle image analysis methods we created. It was seen the detailed profiles of the separation kinetics of lysine and cysteine labeled proteins spanning tens to hundreds of kDa. Results and discussion. Device design fabrication. The on-chips STS page design consists of offset double T-junctions and injection arms to the separating channel. Adjustments to the macrochannel dimensions were made to accommodate single molecule sensing capabilities throughout the separation process. Importantly, the channel depth was made only 650 nanometers deep to ensure single protein to remain in the field of view of a high magnification microscope objective. The low profile channel and also significantly reduces the probability of having more than one protein at each diffraction limited spot at the same time. The macrochannels are connected to 4 liquid reservoirs or ports by through holes etched through the silicon substrate. On the backside, a single port accepts plastic tubing for applying negative pressure, controlled by an electric valve to load protein molecules. An additional 2 ports connect to platinum electrodes for applying voltages up to 30 volts across the 5 mm channels. A 4th port is for loading protein samples, glass sealing by anodic bonding, as well as backside PDMS bonding to create sample reservoirs, and an interface for tubing and electrodes were performed as described previously. For imaging, a wide field microscope consisting of an expanded 641 nm laser iFlex 2000 through a high NA 1.45, 60x oil objective Olympus Plan Apo was used, and images were acquired on an EMCCD camera and/or Ixon 887 at 19.31 frames per second. That's faster than the normal 16 frames, and 1x1 binning to yield a 267 nanometer size. This is the diagram for that. A low profile silicon based fluidic device design for protein separation, lithographic pattern for 10 by 10 millimeter squared chip, including 50 micrometer wide macrochannels on the front side, green, connected to ports exposed on the back side, gray, and subsequently etched through the thickness of the chip by KORAH etching. The depth of the microchannel, 650 nanometers. The left and right ports are for connection to ground and positive electrodes, respectively. The top port is for connection to the vacuum line, and the bottom port is for sample loading. The length of the separating channel is 3.6 mm. B. Schematic of setup showing the optical pathway and vacuum and voltage amplifier connections. A 641 nanometer laser is expanded by a lens L1, L2 and focused onto the chip. The wide field image is filtered by a long pass filter, LPF, and captured on an EMCCD. TL tube lens, Bayes beams filter. C left zoom in of microchannel design showing offsets double T junction separated by 150 micrometer center to center. The interface of the UV exposed polyacrylamide gel is further magnified. On the right side, view of the PDMS layer of the device, back view to facilitate electrodes and tubing connections. The microchannels were prepared for UV activated polyacrylamide gel polymerization using a 2-step channel coating procedure. Microchannels were first functionalized using acrylamide in order to minimize non-specific sticking of proteins to the channel walls as described in methods and channel coating. So let's just check what that methods and channel. Methods, protein labeling. Recombinant proteins, sigma aldrich, were incubated at 37 degrees centigrade for 0.5 to 1 hour in freshly prepared and degassed reaction buffer consisting of 100 micro HEPES sodium hydroxide, 150 sodium chloride, and 6 guanidinium hydrochloride GuHCl, pH of 8.3, Atto6 for 7 NHs. Ester dye was added at a ratio of 1:1 dye. Lysines for bovine serum albumin. of albumin OA and carbonic anhydrase CA, or at a ratio of 2:1 for lysozyme L-wires. And NHS ester is reactive against primary and secondary amines, such as the N-terminus of a protein A amine and the side chain of lysine E amine. GUHCl has a guanidinium group similar to the side chain of arginine, in which the electrons are delocalized among the 3 nitrogen atoms, which provides chemical stability to this group. The proteins were labeled overnight O by N at 25 degrees centigrade with 300 RPM shaking. Subsequently, the proteins were pelleted using TCA precipitation to remove the GUHCl and washed by acetone on ice. The labeled protein pellets were dried and resuspended in 2% SDS, 0.5 mm TCEP in 1,5-phosphate buffersaline PBS for 30 minutes at 37 degrees centigrade with shaking at 300 RPM. Whole cell protein extracts were prepared from HCT 116 cells grown up to 80% confluency to a DME media containing 1 mm sodium pyruvate and 10% fetal calf serum in a humidified chamber at 37 degrees centigrade. The cells were trypsinized, harvested, and washed 3 times with PBS. 2 by 10 raised to 6 cells were resuspended in either lis or cis labeling buffers consisting of 10 mm HBE is sodium hydroxide, 150 mm sodium chloride, and GUHCl at 8.5 or 7.2 respectively. The cell suspensions were sonicated to further disrupt the cell membrane and induce cell lysis. The lysates were classified at 20,000 G for 30 minutes at 4 degrees centigrade. Half of the lysate volume, lysate of 10 raised to 6 cells, was used for each labeling reaction. Each lysate was diluted 1:1 in its appropriate buffer. The cis labeling buffer was adjusted with 40 mm TCEP, and the lysates were incubated for 1 hour at 37 degrees centigrade. Then each lysate was subjected to incubation O by N at 25 degrees centigrade without dye, with 1 mm of Atto 647N NHS ester lysine labeling, or with 1 mg of Atto 647N maleimide cysteine labeling. Subsequently, the labeled lysates were precipitated using TCA precipitation, followed by 4 washes with ice cold acetone. The protein pellets were dried and resuspended in 2% SDS, 0.5 mm TCEP in PBS, shaken at 1000 RPM for 30 minutes at 37 degrees centigrade. Samples of labeled recombinant proteins or whole cell extracts were either subjected to SDS page analysis, followed by fluorescence imaging using the laser gel scanner, Farrow's Biorad, and subsequent Kumashi blue staining, or subjected to single molecule analysis. Prior to the latter, the proteins were denatured in 25 mm tris HCl, 5 mm TCEP, and 2% weight by volume SDS at 90 degrees centigrade for 5 minutes. Proteins were subsequently diluted in running buffer 25 mm tris HCl, 0.25 glycine, and 0.1% weight by volume SDS to 0.1 to 0.4 nm. Chip fabrication. The chip fabrication followed the description by Rehan et al. Ensured low stress silicon NX 200 nm was deposited onto a starting silicon NX by silicon dioxide silicon wafer 50 nm by 350 nm by 350 micrometer by PECVD 300 degrees centigrade in order to increase the workable channel depth. Following cleaning in solvents, acetone, methanol, isopropanol water, the water was baked on a hot plate at 300 degrees centigrade for 10 minutes. AZ1518 resist was spin coated at 4000 RPM to a thickness of 1.8 micrometer and baked on a hot plate at 120 degrees centigrade for 2 minutes. The macrochannels were exposed by UV light. MacroWriter ML3 developed in Nova Developer 2.14% TMAH water, subsequently etched by reactive ion etching RIE with 10 SCCM CF4 and 10 SCCM O2, 75 watts 0.15 m bar. Next, 0.5 mm through ports were exposed as individual squares, backside aligned to the macro channels, along with cut lines. The SiR-NET and underlying SiO2 insulating layer were etched by RIE and BOE, respectively, and then opened by anisotropic etching. Then 33% KOH for individual 10 by 10 millimeter square chips. Channel coating. Channels were cleaned by an initial flush with 1M NaOH for 10 minutes, followed by flushing with deionized DI water. Subsequently, the channels were conditioned for acrylamide coating by incubating with 2:3:5 mixture of 3-trimethoxysilyl propyl methacrylate, glacial acetic acid, and deionized water for 30 minutes, and then rinsing with methanol and deionized water for 10 minutes each. A solution of 5% weight by volume acrylamide coating containing 5 mg by ml, 2% azobis 2-methylpropionamide dihydrochloride V50 photo initiator was flushed through the channels, and the channels were then exposed to 365 nm UV light in close proximity, less than 3 mm for 10 minutes. Spectroline ENB 260C. Chymographs construction. The chymographs were constructed by concatenating the central pixel column of each frame, column 256, from a fixed location along the channel, 1.3 mm for the 1.5 mm gel and 2.3 mm for the 2.5 mm gel. The pixel values represent the intensity values obtained from the EMCCD camera. The dynamic range of the image is then adjusted to produce a clear image of the labeled proteins. Protein detection algorithm. The original accumulated image before rescaling pixel values is filtered using a Laplacian of Gaussian LOG filter, which results in image segmentation where each segment represents a single protein. The segment center of gravity corresponds to the protein location in the image, and the total energy, pixel value, and the area correlates with the protein intensity. The protein's time of arrival is determined by the center of gravity location along the horizontal axis divided by the frame rate. The protein's intensity is the normalized value of the segment energy. Coming back to SMMDs, the rationale for developing SMMDs. Micron scale measurements of molecular diffusivity have proven to be a versatile and sensitive approach for probing the molecular sizes of proteins, their interactions and assembly states in solution. In particular, microfluidic diffusion sizing MDS has become an attractive quantitative method for characterization of proteins, protein complexes under native solution conditions. MDS exploits the unique features of laminar flow in the macrofluidic regime and measures the diffuse mass transport of molecules across co-flowing sample and buffer streams within a microchannel. By monitoring the diffusive spreading of analyte molecules at different downstream channel positions and analyzing the recorded diffusion profiles with advection diffusion models, the size of analyte molecules can be quantified in terms of their hydrodynamic radii RH. Importantly, MDS offers the advantage of being calibration free as it directly retrieves RH from model fitting, thereby eliminating the need for external calibration standards. MDS can further probe the formation of biomolecular interactions and the assembly of analyte molecules into higher order structures by monitoring the increase in size associated with complex formation, and can retrieve binding affinities, that is dissociation constant, KDS, through measurement of binding curves. However, despite the versatility of the approach, current implementation of MTS and other similar microfluidic methods are limited in their ability to resolve compositional heterogeneities. This is because they rely on ensemble readouts that average out the sigma, making it difficult to determine the size distribution of different species, thus yielding only an average RH for mixtures of differently sized species. This limits information content, particularly when studying heterogeneous multi-component systems. Additionally, detection sensitivities for MDS And other similar sizing techniques are in the nanomolar to macromolar range, which hampers ultra-sensitive protein detection at concentrations in the often desirable pico to femtomolar range. A more sensitive detection method like confocal fluorescence detection could offer an effective solution to these challenges. This technique enables ultra-sensitive detection at the single molecular level, allows for direct digital readouts by counting individual proteins and protein complexes in solution, and can be seamlessly integrated with the macrofluidic platforms, thus setting the stage for developing a single molecule digital sizing method. And it has been reasoned that combining single molecule fluorescence detection with diffusivity measurements in macrochannels would create a robust platform for sizing proteins at the single molecule level in a calibration-free manner. Such a platform would be ideally suited for characterizing heterogeneous and multi-component protein systems directly in solution. Working principle and experimental implementation of SMMDs. The figure 1.8 illustrates the working principle and experimental implementation of a SMMDs, which measures the molecular diffusivity of analyte molecules within a microfluidic chip. It operates based on the principles MDS and probes molecular diffusivity by flow focusing an analyte stream between two auxiliary buffer streams within a microfluidic chip, and then observing the diffusive spreading of analyte molecules to either side of the microfluidic channel as they travel downstairs. Because different positions along the channel correspond to different diffusion times, the tracking of the diffusive broadening of species at different channel positions allow calibration, free quantification of the diffusion coefficient, D, and thus extraction of the size of analyte molecules in terms of RH. Experimentally, SMMDs measurements are conducted by introducing a sample containing fluorescent protein and buffer into the sizing chip and monitoring the micron scale diffusive mass transport of molecules across the channel as they flow downstream the channel. Fluid flow in the channel is controlled by applying a negative pressure at the device outlet with a syringe pump. This is a diagram showing a working principle and experimental implementation SMMDs. Now, detection in SMMDs is achieved using a high sensitivity laser confocal fluorescence, which was already mentioned. Microscope functionality incorporated into the macrofluidic platform, as seen in the methods. By scanning the confocal volume across the macrofluidic chip at the mid-height of the channel perpendicular to the flow direction, fluorescence from passing analyte molecules is recorded. The scan trajectory is chosen such that various positions along the channel are probed, including positions that are close to the nozzle where the sample stream meets the co-flowing buffer medium, and others further away downstream of the channel. In the implementation, the four innermost channels of the device were scanned to obtain diffusion profiles. These selected channels cover a wide range of distances and time points along the channel's length. This enables analysis of biomolecular analytes with RH ranging from less than 1 nanometer to greater than 100 nanometer, paralleling the established range in standard MDS experiments. The scan trajectory of the confocal volume in XYZ direction is set through two scan markers integrated with the microfluidic chip adjacent to the channels. Scanning is conducted in two modes, by either continuously moving the confocal volume through the chip, or by the observation volume along the same trajectory in a stepwise manner, collecting data at defined positions with each step. In continuous scan mode, diffusion profiles are rapidly acquired from direct intensity readouts. In this process, the confocal volume is moved through the device at a constant scan speed 10 per micrometer per second, and the fluorescence intensity from analyte sample flowing through the confocal volume is recorded. This allows swift recordings of diffusion profiles under ensemble conditions, that is, at concentrations where many molecules are present in the confocal volume, that is typically at concentrations greater than tens of ppm. An exemplary diffusion profile obtained from a continuous scan experiment is shown in Figure 1b. To extract RH, the recorded diffusion profile is analyzed using an advection diffusion model, as shown in methods. This process involves fitting the experimental profiles to a set of simulated profiles generated through numerical simulations. Using at least squares error algorithm, the best matching simulated profile is identified via fitting to extra D and retrieved using the Stokes-Einstein relationship. In step scan mode, diffusion profiles are generated from time trace recordings along the scan trajectory. An exemplary diffusion profile obtained from a step scan experiment is shown in Figure 1C. In this process, the confocal volume is popped at various positions along the trajectory, and fluorescent signals from analyte flowing through the confocal volume are recorded for a certain period of time. Sizing of proteins from bulk to single molecule conditions by SM-MDS. In the first set of experiments, it was to evaluate the sensitivity of SM-MDS and demonstrate its capability to determine protein size from bulk to single molecule conditions. To this end, human serum albumin was labeled HSA with a fluorescent dye LX488 as shown in methods and supplementary table, and perform concentration series measurements both in continuous and step scanning mode by varying the HSA concentration. Recorded diffusion profiles of the series as shown 2BC. Sizing of proteins from bulk to single molecule conditions by SM-MDS. The sensitivity of SM-MDS and its capability to size proteins from bulk to single molecule conditions was evaluated by measuring the size of human serum albumin HSA at varying protein concentrations. Diffusion profiles of HSA as obtained from continuous scan measurements from top to bottom. In the range from 1 micrometer down to tens of picomoles of HSA, sufficient molecular flux of HSA proteins molecules allowed for the recording of diffusion profiles from continuous scan experiments. The obtained profiles show the characteristic broadening due to diffusion of molecules along the channels. Narrow peaks are observed at channel positions close to the nozzle where the sample meets the carrier medium, and peaks broadened as it was probed further downstream channel positions. Modeling of the diffusion profiles using advection diffusion analysis approach yielded excellent fits. Extraction RH values were among all concentrations with an error in excellent agreement with the previously reported values for HSA, demonstrating the robustness and accuracy of the approach. Of note, in some cases, the diffusion profiles display a minor offset from the channel centers. It is attributed to potential small imperfections upstream in the flow path, which can slightly shift the peak positioning. However, the global fitting procedure utilized for extraction diffusion constant is resilient towards anomalous shifts that occur within channels, ensuring that overall analysis remains robust and accurate. The single molecular regime, that is at and below 20 picomoles, performed step scan measurements by moving the confocal volume in a stepwise manner across the channels. It was observed that there were bursts of fluorescence corresponding to the passage of single HSA molecules through the confocal volume, and using the burst search algorithm as shown in methods. Let's have a look at the burst search algorithm. And single molecule events from the record time trace at each position, and created diffusion profiles by plotting number of countless molecules as a function of chip position. Hence, HSA exists in the equilibrium of monomers and low-order oligomers, which are populated with decreasing relative abundance. Hence, RH values reported are weighted averages reflecting this distribution of the monomeric oligomeric forms for deconvolution analysis. On the whole, results shown here demonstrate the SM-MDS provides accurate size information over a broad range of concentrations and enables ultrasensitive sizing of proteins even in the picomolar to femtomolar concentrations regime. It is also evaluated that the influence of parameter selection in the single molecule regime on the extracted sizes, and found that a wide range of burst selection parameters, that is varying thresholds of ITP max and NMIN, yield the expected size information supporting the robustness of the approach. To compare the sensitivity of SMMDS technique to conventional MDS measurements, experiments utilizing fluorescence, wide field imaging were conducted, and concentration series measurements by varying the HSA concentration starting at 1 micro mole of labeled HSA and then gradually decreasing protein concentration down to 100 to 50 nm. That's 100 and then down to 50 nm. Image analysis yielded a clear profile only for measurements at 1 micro m and 100 nm protein concentration. An expected size for HSA could only be recovered for the measurement at 1 micro m protein. Notably, the measurements at 50 nm HSA yielded a featureless profile that could not be fitted, and hence no RH could be determined. Sizing of proteins and protein assemblies by SMDS. The wide applicability of SMMDs in determining the size of proteins and protein assemblies from single molecule digital counting was done. A varied set of analytes differing in size includes the proteins, lysozyme, RNase, alpha-synuclein, human leukocyte antigen HLA, HSA, thyroglobulin, and oligomers formed by the protein alpha-synuclein. These protein analytes collectively span a size range of 1 to 10 nanometer. Also included were the small organic fluorophore LX488 in the series as a sub-nm sized analyte. The protein analytes were fluorescently labeled and purified before analysis. To demonstrate the detection of biomolecular interactions and quantification of binding affinities by SMMDS in a digital manner, the binding of clinically relevant antibody-antigen interaction was done. Specifically, the binding interaction between HLA A03:01, an isoform of the major histocompatibility complex type 1 MHC, and a key factor in the human immune system, and the antibody W6/32, an antibody that binds to all class 1 HLA molecules. And a series of step-scan SMDS experiments were conducted by titrating HLA antigen labeled with LX488 at a constant concentration of 100 picomoles with increasing amounts of the unlabeled W6/32 antibody. And the labeled HLA and unlabeled W6/32 antibody in the study, motivated by the clinical significance of detecting anti-HLA antibodies in patient serum, especially in scenarios involving organ transplantation. Exemplary diffusion profiles of a few HLA at 100 picomoles and 100 picomoles. HLA treated with 10 nm of W6/32, shown in the figure as SMMDS diffusion profiles from 3 repeats were acquired and failed to obtain effective RH across the concentration series. It was observed that there was an increase in average hydrodynamic radius from RH, 3.18 plus or minus 0.04 nanometer for pure HLA, corresponding to a molecular weight of 50 kDa as expected for HLA, to RH 5.08 plus or minus 0.01 nm for saturated complex, corresponding to the molecular weight of 215 kDa, consistent with the binding of 150 kDa antibody to HLA. Importantly, HLA antibodies are an extensively used clinical biomarker to evaluate, for example, histocompatibility and the risk of allograft rejection. Given that these antibodies are found in patient serum at very low concentrations, our findings offer a method for detecting and profiling antibody responses even when only minimal sample quantities are available. Quantifying protein interactions by SMMDS. Sizing and affinity measurements of an antibody-antigen complex by SMMDS, shown as a schematic for the binding interaction between human leukocyte antigen and the human HLA antibody. Exemplary diffusion profiles for binding of HLA antibody W6/32 to HLA as obtained by step scan SMMDS. Overall, the results were highlighted, showed the potential of quantifying biomolecular interactions through single molecular digital measurements, even at very low concentrations, and the possibility to measure at very low protein concentration enables examining high affinity interactions. Resolving protein oligomeric states by SMMDS, sizing of heterogeneous oligomer populations by SMMDS, sizing of multiple species within a heterogeneous aggregation mixture by SMMDS, sizing of nanoscale clusters by SMMDS, and discussion. The physical characteristics of the sizes, interaction, assembly states of proteins is vital for a heightening understanding of biological function, malfunction, therapeutic intervention of proteins. Of particular interest are insights into the composition, heterogeneity of proteins and protein mixtures such as protein oligomers, protein aggregation, and protein condensate systems. The rest will be continued in the next lecture. Thank you.